Greetings of the day. Today we are going to see about the first week of the development. In the last week already we have seen the formation of the gametes, that is the developments of the sperm and ovum, which are including different processes. So today in this video we will be seeing about the first week of the development, so which includes the different activities. So let us name one by one. So first is the fertilization, and second is the cleavage, and third is the blastocyst formation. So these are the three important activities we will be seeing today. So first of all, in the last video, as I have discussed about the gametes, the next process will be happening in the life for the development of the life is the fertilization. So the just to define the fertilization, that is the fusion of the male and female gametes in a single line definition, that is the fusion of male and female gametes is called as the fertilization. So after this one, the important site, or you can say the site of the fertilization, is the ampulla of the fallopian tube. Usually asked MCQ for the first year, that is the ampulla of the fallopian tube, which is the actually most dilated part of the fallopian tube, is the site of the fertilization. And after the fertilization, the product formed is called as the zygote, which is actually formed due to the fusion of two pronuclei. So in the last video, as we have seen the oocyte formation and the sperm formation, so there occurred a formation of the male pronucleus and female pronucleus, which will be fused, and then the product formed due to that fusion is called as the zygote. So now this is the definition for the fertilization. Now after this one, actually events causing due to the fertilization are mainly, first of all, as soon as the fusion happens, immediately the sex of the fetus will be determined. So that is one of the event, first important event will be happening as soon as the zygote is formed. And next one, immediately the restoration of the diploidy. Why? Because in the last video we have discussed about the ploidy of the gametes, or the ploidy of the sperm and the oocyte. If just we recollect them, they both are the haploid. Both sperm and ova both are haploid in their own nature, and now due to this fusion, these both will be forming the fused, and then formed the diploidy will be restored. And lastly, the next important event is the initiation of the cleavage. So this is how the events of the fertilization can be explained. So when the question comes, define fertilization. So first of all, the fertilization is the fusion of the male and female gametes, and the site of the occurring, that is the ampulla, which is the most dilated form, due to which we have seen the formation of the zygote. So these are all the important points to be understood at this level. What is the meaning of the fertilization definition, and what are the events happen followed by the fertilization, and due to the fertilization the sex of the fetus is determined at that level only, and after that one, restoration of the diploidy, and then the initiation of the cleavage. So now we will see about these steps in detail. So now let us try to understand what are the steps here. So the approximation of the gametes, the approximation of the gametes has to occur first of all, or coming nearer of the gametes. So how these gametes will be coming nearer to each other? So after the coitus, what happens is that approximately or usually 200 to 300 million sperms will be deposited in the female genital tract, of which only one will be successful. So that is the only one will be successful, and that only will be fusing with the ovum, so that the zygote will be formed. So now now the point to be noted is that why this much high number? Why? Because the important point to be noted is that mortality of the sperms in female genital tract is higher. So that is the reason why the number of sperms per ejaculate will be more. Okay, and after this one, and only 200 to 300 sperms will be reaching the oocyte. So the journey of the sperms from the vagina to the ampoule of the uterine tube has to be understood. What are the factors responsible? Factors responsible for the transport of the sperms. So factors responsible for the transport of the sperms are the, first of all, prostaglandins, that is promoted by an important chemical called as the prostaglandins, which induce the contractions, so that the sperms will can be moving forward. And now where is the source of the prostaglandins? They are from the semen only. So prostaglandins are from the semen, and then they cause the powerful contraction, as I have said already. And also prostaglandin assisted by or associated by the oxytocin from the pituitary gland, and uterine contractions, they cause a negative pressure. So after the prostaglandins plus the oxytocin and also the uterine contractions, these are all the factors responsible for the sperms from the vaginal cavity into the uterus, and from there into the ampulla. So from the cervix to the isthmus of the uterine tube, they occur due to their own propulsive action, due to their whipcord like action. And now the important timeline or the important period of the time is 2 to 7 hours. 2 to 7 hours is required for the transport of the sperms from the cervix to the ampulla. So 2 to the 7 hours is the time required for the transport of the sperms from cervix to ampulla. And then after this one, what happens is that as it is already discussed, of the all 200 to 300 million sperms, only 1% of the sperms will be reaching the uterine tube at the time of the fertilization. So this is the first level to be understood, that is the approximation of in the approximation of the gamete. The first part is the movement and transport of the sperms. Now in that first step only, the second step is the movement of the oocyte. As such, movement of the oocyte is immotile. Actually, the point to be noted is that oocyte is immotile, whereas the sperms are motile. So transportation of the oocyte, you can say, that is what happens is that just before the ovulation, the fimbriated end of the uterine tube. So in a simple diagram to be noted, so this is the fallopian tube, and at the lateral end of the fallopian tube there will be a fimbriated end, and here it will be the ovary. And in the, as I will said the last video, the oocyte will be forming an surface bulging on the ovary, and this fimbriated end of the fallopian tube will cover that region and take the oocyte into it, so that the oocyte will be transporting in this direction, whereas the sperms are coming in this direction. So this is the uterus, and this is the other side fallopian tube. So this is the vaginal cavity, and the sperms which are deposited here, they follow in this direction. So this is actually the uterine cavity. So the sperms are forming in this direction and the oocyte is coming in this direction. So that is how the oocyte will be transported. And now how does the oocyte is moving? The point to be noted, as I have said, oocyte itself, it is immotile. Then how it is moving is that it is a passive movement. To understand that it is a passive movement, and that action is called as the sweeping action. So the finger like fimbriae start moving back and forth, that is the sweeping action. And then from the and then into the infundibulum of the uterine tube as soon as it is discharged from the ovarian follicle. And from the infundibulum, what happens is that the oocyte passes to the, so here this is the infundibulum. So first one, infundibulum, and here this is the ampulla. So here it goes, and then sperms will be moving it towards that one. Okay, so after this one, the sperms again become motile, more motile, due to the chemoattractants produced by the cumulus cells surrounding the oocyte. So what happens is that the sperms which are deposited, once again they will become motile due to the chemoattractants produced by the oocyte cells, and they get attracted, and due to which they will be moving towards the oocyte for the fertilization. And the majority of the sperms die within the 24 hours. So this is about the movement of the oocyte and also the movement of the sperms. Okay, so then after this one, the point to be noted is that the viability of the gametes. So the viability of the gametes will help in determining the time of the fertilization, so as the couple can achieve the success result. So after this one, the point is that the viability of the gametes. So the viability of the gametes is actually the female gametes, the mature oocytes. The mature oocytes will be viable for the 24 hours. So oocyte viability is 24 hours after ovulation. 24 hours after ovulation, and after usually fertilized after 12 hours after the ovulation. And now male gametes, so that is the sperms, what happens is that the sperms will be motile, sperms will be viable for 48 hours in the female genital tract. If they are in the female genital tract, they will be viable for the 48 hours. So that is this is the viability of the gametes. And the next point is that the fertilization barriers. So as such, the fertilization is not a smooth procedure. And why? Because the for the fertilization to happen, there are a three number of the barriers, or there are three barriers to be crossed. So one by one, let us try to understand. So the fertilization barriers to be understood is that the fertilization barriers. So first of all, that is formed by the corona radiata. So in the last video, when I said you, that is the corona radiata is the group of the cells, 2 to 3 layers, that is formed by the follicular cells surrounded by, so corona radiata usually 2 to 3 layers, and the source is by the follicular cells surrounding the developing oocyte, follicular cells. And after this one, next one, that is the second barrier, that is the zona pellucida, which is actually a glycoprotein, which is actually a glycoprotein, and it is a thick membrane of the same glycoprotein surrounding the oocyte deep to the corona radiata. So this is the first barrier, this is the second barrier, and then the third one is the vital line membrane of the oocyte itself. So vital line membrane of the oocyte, vital line membrane of the oocyte. So these are the three barriers, and the zona pellucida, which has got a very importance, and usually asked as the 3 mark section. So what is actually the zona pellucida, and there are some important functions for the zona pellucida. So let us try to understand one by one what are the functions of the zona pellucida. And actually the zona pellucida, what does it do is that it protects the oocytes, it protects the oocytes and prevents the implantation at the abnormal site mainly. So protects the oocytes and early embryos. And next one, second one is that the diffusion of the uterine secretion. So what is happening is that in the initial days the nutrition for the early embryos has to be done by the endometrial cells lining the uterus. Okay, so that is, there is a special name for that, separate name for that one, that is called as the witch milk. Okay, so diffusion of the uterine secretion, diffusion of the uterine secretion, which has got a separate name, that is called as the witch milk. The point to be understood is that at this level the uterus will be in the secretory phase of the menstrual cycle, and then after this one the third function is that regulation of the interaction between the sperms and the oocytes, regulation of the interaction between the sperms and the oocytes during the fertilization. And after this one, due to this binding of the proteins, it evokes a separate reaction called as the acrosomal reaction, which is another 3 marks important question. So after this one, what happens is that the preventing the polyspermy. So the meaning of the polyspermy is that once one sperm breaks the barriers and enters the ovum, it prevents the another sperms to once again break open the barrier, so that polyspermy subsequently that twinning, all these cleavage malformations in the cleavage can be separated, can be avoided. So that is the polyspermy, polyspermy is prevented. And most importantly, last but not the least, is that abnormal ectopic implantation, abnormal or the ectopic implantation is prevented by the zona pellucida. So these are all the important functions of the zona pellucida. So, and of all these, the same as I have said, it prevents the ectopic implantation. So that is now due to which now the for the sperm to fertilize the ovum, these are the three barriers. First is the corona radiata, so just that is the 2 to 3 layers of the follicular cells surrounding the developing oocyte. And zona pellucida, it is basically protein in nature and it has got very important functions, and so that's what we have discussed. And next one, that is the vital line membrane. So after this one, so how can a sperm cross these barriers? Is the sperm that much powerful enough to cross these barriers? Okay, yes, so let us discuss about this one. So what happens is that the sperms gets this ability, definitely sperms gets this ability. So how is that? Is that it undergoes two processes. So one is the capacitation, and another is the acrosome reaction. So one is the capacitation and another is acrosome reaction. So these are the two important processes to be undergone by a sperm so that the fertilization barriers are crossed. So just give me a moment so that we will discuss about the capacitation acrosome reaction in detail first, and next we will be understanding the stages of the fertilization in a sequence wise. So in that next one, let us understand about the what is mean by capacitation. Once again a 3 marks question here. The that is the what is mean by the capacitation. So capacitation is defined as the, or else before going the definition, that is the final step of the maturation. So let us understand, that is the final step of the maturation for the sperms. So final step of maturation for the sperms to fertilize the oocyte. And next one, that is the what is that is that where does it occur? It usually occurs in the female genital tract only, and this process will be happening for about 7 to 10 hours. The point to be understood and point to be remembered is that only after undergoing the capacitation a sperm can fertilize the ovum. So during the capacitation, what is the actual process being happen? Okay, so the during capacitation what happens is that the glycoprotein coat and the seminal plasma proteins, they which are covering the plasma membrane of the sperm, are removed. Okay, so that means, so to simply sum up, what happens is that the glycoprotein and the seminal plasma protein, so glycoprotein coat and so glycoprotein coat plus seminal plasma coat, seminal plasma coat, both are lost, both are lost. Okay, so that is what is the main important chemical process happening in the capacitation. And who are removing? They are removed by the chemicals secreted by the uterus, due to which, so after which what happens is that the tail starts wagging more, tail of the sperm starts wagging more, wag more. So tail of the sperms wag more, and now due to which, as I have said already, only capacitated sperm undergoes the acrosome reaction, and after undergoing the acrosome reaction, that is how it receive, how it gets the energy to fertilize the first step we have seen. And now next one, that is the acrosome reaction. So usually asked 3 marks question, that is the one is the capacitation and another is the acrosome reaction. So acrosome reaction happens only after the capacitation. So what is happening is that when capacitated sperm undergoes the, or else it comes in contact with the zona pellucida. So that means a capacitated sperm has to undergo the contact, or has to come in contact with the zona pellucida of the oocyte, zona pellucida. So after this one, what is happening is that, that too particularly to specifically say, acrosome reaction is induced by zona proteins only. Okay, so this reaction, due to which what happens is that, due to which the hyaluronidase and acrosin will be released. Okay, so let me write it. So hyaluronidase and acrosin are released. Okay, so the source is that from the acrosomal cap. So these two are released from the acrosomal cap, due to which the acrosomal reaction takes place. Okay, and after this one, what is happening is that the antigenic coating of the sperm initiates an immunological reaction, so between the oocytes fertilizing and sperms anti-fertilizing, or simply you can say acrosome reaction is the one which happens when a capacitated sperm. So while writing, it should be written like that, only the capacitated sperm. So just writing the sperm won't suffice the answer. Why? Because the processes are in a chronological order. So first capacitation has to happen, and then only it can undergo the acrosome reaction. So when the capacitated sperm comes in contact with the zona pellucida, immediately the hyaluronidase and acrosin are the chemicals which are released from the acrosomal cap will happen, and this entire process is called as the acrosome reaction. Now we are moving into the important step, that is the fertilization, and which includes around some 7 to 8 steps clearly, so that we will discuss now. So let me write the first of all stages of the fertilization. Stages of the fertilization. So these stages of the fertilization, as we have seen, it has to break the three barriers, so maybe called as the fertilization barriers. And now the sequence of the events taking place during fertilization will be, so let us write one by one. So that is the penetration of the corona radiata. Penetration of corona radiata, which is surrounding the developing oocyte. So next one, that is the penetration of the zona pellucida. Penetration of the zona pellucida. So next one, that is the penetration of the vital line membrane. So this already we have discussed. So these are the three barriers of the fertilization. So penetration of the corona radiata, penetration of the zona pellucida, and penetration of the vital line membrane, and after which happens the formation of the pronucleus, female pronucleus, formation of the female pronucleus. So after this one, formation of the male pronucleus, formation of the male pronucleus, and after this one, the next one, that is the formation of the zygote, formation of the zygote. So as it is important, so that is the formation of the zygote. And after this one, so these are all the important stages of the fertilization. So let us discuss about each one in detail. So usually fertilization will be asked in the 6 marks or the 5 mark section depending upon the place. And now here this is how we have to start the answer with. So first of all, these are the fusion of the male and female gamete is called as the fertilization, and different stages of the fertilization are to be written in a line wise, and we have to explain it. So penetration of the corona radiata. So now here the penetration of the corona radiata, in that first one, let us see about 300 to 400 sperms. So I am not saying the million, so here it is only 300 to 400 capacitated sperms. As I have said you already, capacitated, that word has to be repeated. So capacitated sperms will come into contact with the corona radiata, and they liberate the, or they release the hyaluronidase enzyme. So that's what we have seen, what is mean by the acrosome reaction. So after this one, what is happening is that due to which what happens is that the chemical binding the cells of the corona radiata. So chemical binding the cells of the corona radiata will be digested. So chemical binding cells of corona radiata are digested. So this is the first level. So that is the penetration of the corona radiata. Now what is happening is that the first barrier, it's got broken, that is the corona radiata. So to reach the zona pellucida. So that is the next one, that is the next is the penetration of the zona pellucida, ZP. So that is the what, let us try to understand what is mean by the what is happening. So what happens is that the just few moments back what I have discussed is the contact after the glycoproteins of the zona pellucida come in contact, an acrosome reaction happens. So acrosin and pepsin like substances gets released, and then the zona pellucida also gets lysis or breakdown. Okay, so due to which the second barrier also is cleared, and now it reaches the perivitaline space. Okay, so once the sperm penetrates the zona pellucida, a change in the substances happens, which is called as the zona reaction, and due to this zona reaction, it becomes impermeable to the other sperms to prevent the polyspermy. So breakdown of the chemicals, or breakdown of the zona pellucida membrane. breakdown of the zona. And now next one, that is the third, that is the penetration of the vital line membrane. Penetration of the vital line membrane. So when the plasma membranes of the sperm come in contact with that of the plasma membrane of the oocyte, which is the actually vital line membrane. So vital line membrane can be understood as the plasma membrane of the oocyte is nothing but the perivital line membrane. So plasma membrane of the sperm comes in contact with the plasma membrane of the oocyte. So due to which what happens is that at the site of the fusion, this allows the head of the sperm to enter into the cytoplasm of the oocyte, leaving behind the body. And now important point here is that as soon as the head and neck of the sperm enters, a calcium wave appears in the cytoplasm of the oocyte. Calcium wave, where does it is happening? In the cytoplasm of the oocyte. So who is triggering this calcium wave of the calcium wave in the cytoplasm of the oocyte? When the head and neck of the sperm enters the breaks the vital line membrane. Head and neck of the sperm enters the oocyte. It induces a calcium wave in the cytoplasm of the oocyte, and this is also one another reason the other sperms will become cannot permeate the ovum. So this is the next level, that is the penetration of the vital line membrane. So after this one, what happens is that the formation of the female pronuclei will be happening. Okay. So now the next important step will be the formation of the female pronucleus. So in the last video, in the previous week's video, we have seen about the gamete, and there the which also includes the female pronucleus and male pronucleus. So now here the female pronucleus, what happens is that the penetration, so the penetration of the oocyte, or the penetration of the sperm activates the oocyte to complete its second meiotic division. So this is the important step. The second meiotic division is induced by penetration of the sperm head and neck. Now what is happening is that the second meiotic division is completed, and due to which the two cells are produced. So in the second meiotic division, the results are a bit interesting to be noted. So as I said, two cells are produced, and one cell contains all the cytoplasm. One cell contains all the cytoplasm, and the other cell contains only the or else you can say hardly any cytoplasm. So one cell contains all the cytoplasm is called as the second polar body. In the previous video, in the gametogenesis, we have seen the formation of the first polar body. Now here the second polar body is formed, and now other cell hardly contains cytoplasm. So all the cytoplasm was gone with the second polar body. Now only hardly any cytoplasm is left, and that is the other cell. And what does it contain? That only the chromosomal content. So that is the 22 plus X, that is the genetic makeup of this one, of the female pronucleus, and they arrange and condense themselves in a vesicular pattern. Now after this one, next one, that is the formation of the male pronucleus. So just before we have seen the female pronucleus, now that is the male pronucleus. So now The sperms move forward to come in close contact with the female pronucleus, and the same, just the sperm's nucleus will become swollen and forms the male pronucleus. And morphologically, the male and female are indistinguishable or can't be identified easily. So each chromosome in female and male pronuclei is made of only one chromatid. And now the oocyte containing two haploid nuclei is called as the oocyte. So there is a separate name we have seen, that is the oocyte. So what is the oocyte? Is that it is the oocyte with two haploid nuclei. So with a male haploid nuclei and female haploid nucleus, sorry. So oocyte with two haploid nuclei, that is the male haploid nucleus and female haploid nucleus, that is called as the oocyte. Now after this one, formation of the zygote. Formation of the zygote. So formation of the zygote, once the after the fusion, the male and the female pronucleus, they will lose their cell membrane, and the chromosomes, they mix together to form the diploid. So now the oocyte has become the zygote, and the chromosomes in zygote become arranged on the cleavage spindle. So that is how the cell division happens. And so here in the formation of the zygote, at the at that very micro level, the point to be noted is that they both will mix together. The chromosomes content will be mixing together, and cleavage spindle will be arranged in preparation for the cleavage of the zygote. And chromosomes split longitudinally. Chromosomes split longitudinally. And now this is how the fertilization happens and the zygote forms. Okay, so now the results of the fertilization. After understanding the stages of the fertilization, the point to be noted is that the result of the fertilization. So what are the results of the fertilization? What happens? So first of all, the results of the fertilization includes the completion of the second meiotic division of the female gamete. Results of the fertilization. So the first is the completion of the second meiotic division. So then after this one, restoration of the diploid number of the chromosomes, which is the very important step, that is the restoration of the number of the chromosomes, or you can say just restoration of the diploidy. Restoration of the diploidy. And most importantly, determination of the gender. Determination of the gender. So here what is happening is that there is an important concept here. So there are two types of the sperms which are named accordingly to their chromosome type. So androsomes are there and gynosomes are there. So androsomes and gynosomes. So androsomes are the one which are Y bearing, and the gynosomes are the one which are X bearing. So when an androsome or the Y containing sperm, or the androgenic sperm fuses with the oocyte, then the boy will be born, and when the gynosome is fused with the ovum, then a female baby will be born. Okay, so that is about the result of the fertilization, the third step. And after this one, that is the initiation of the cleavage. initiation of the cleavage, and then variation of the human species. So fertilization stimulates the zygote to undergo a rapid many divisions, so that is called as the cleavage. So that is about the stages of the fertilization. And now here we will see the actual formation of the cell. So that actual formation of the actual cleavage and the blastocyst formation. So for the blastocyst to form, the step to be understood is that the first the cleavage. So the cleavage formation, actually first of all let us try to define the cleavage. So the cleavage can be understood as the simple one line answer: repeated mitotic divisions. Repeated mitotic divisions. So that is what is mean by the cleavage of the zygote into the smaller units. And now due to which what happens is that the cells formed are called as the cells formed due to the repeated mitotic division are called as the blastomeres, or they are termed as the blastomeres, and which become smaller with the successful division. And now the division of the zygotes just starts immediately after the fertilization. Now during the cleavage, what is happening is that the zygote is protected or surrounded by the thick layer, that is the zona pellucida. Now first of all the zygote, let me draw the diagram here. Zygote, so that is the single cell stage, and here so the red color one, that is the zona pellucida. So that is the zona pellucida. And now what happens is that in that the that is the two cell stage is there. So here with their respective nucleus. And now what is happening is that this is the two cell stage. So from the two cell stage what happens is that if you so it can be understood from this diagram that one is larger and one is smaller. Now what is happening is that the larger cell divides first, due to which we will get the three cell stage. So 1, 2 and the 3 cell stage. So this is termed as the 3 cell stage, and due to which what happens is that further it then goes into the 4 cell stage. 1, 2, 3, 4 with the nucleus and then the nucleolus. So this is named as the 4 cell stage. So after this one, due to further continuation, will be resulting in the 16 cell stage, which is resembling the mulberry fruit. That is why it is called as the morula. Okay, so in all this what is happening in all these 4 stages, so the layer now we can show that is the zona pellucida. So in all this zona pellucida will be there. So to recollect, the zona pellucida function is to prevent the abnormal implantation. So here these are this is the 16 cell layer. this is the 16 cell stage, and as it is resembling the mulberry fruit, we will name them that as the morula. And here these are all called as the blastomeres with little cytoplasm for the surviving. So now what happens is that the morula or the the morula, it enters the uterine cavity, so which can be said as the, which can be understood as the blastocyst formation. So now what happens is that till now the one cell will become 2, 2, 3, and then what is happening is that, what happens is that the finally the 16 cell stage is reached, which is called as the morula. And after the formation of the morula, then the blastocyst is formed. So let us try to understand how it is formed. So now, so it is more easily understood with the help of the diagram. So this is the zona pellucida. Let me label it first of all. Now in the 16 cell, what happens is that after the 4 days of the fertilization, the morula enters the uterine cavity and endometrial fluid. Now the important step is that the endometrial fluid, by a process called as the diffusion, enters into the cavity, and due to which, for the nutritional purpose, and what happens is that the intercellular spaces of the morula will be increasing and coalescing to form a single large cavity called as the blastocyst cavity. And now this blastocyst cavity is filled with the, so that I will say you. So first of all, so this in this blastocyst or the 16 cell morula, they will be undergoing into two lines. So that is the first of all outer layer, yes, this is the outer layer with their respective nuclei, and now into which we will be seeing the inner layer. So here the color difference is clear. So outer layer and inner layer of the cells. And now in the next level, what happens is that the outer, as I have said, the endometrial fluid will be entering into this cavity through the diffusion, and then what happens is that it forms a cavity called as the blastocyst cavity, and due to which what happens is that the cells will be migrating to the one end. Okay, so the cells now get arranged into two groups, that is the inner cell mass, and then the cells at the periphery of the morula are called as the outer cell mass. So in the after further processes, what happens is that we will be seeing the two layers of the cells. So this is the outer layer. Okay, so this is the outer layer, and now at this end what happens is that there is the inner layer. So here we can understand that there is a cavity formed, that is called as the blastocoele, or you can say the blastocyst cavity. So the blastocyst cavity is filled with the uterine milk. Okay, so now here we can say, by seeing this diagram, the cells are arranged into two groups, the two, one in the center, that is the inner cell mass, that inner cell mass is also called as the embryoblast. Inner cell mass is also called as the embryoblast. And now the next one, that is the outer cell mass, that is called as the trophoblast. Outer cell mass, that is called as the trophoblast. trophoblast. As the cavity enlarges, now what is happening is that they becomes the different poles. Okay, so now let me draw a magnified diagram here. So now these outer cells of the trophoblast will become the flattened cells. So earlier it is not a flattened, now here it is the flattened cells. So with their respective nuclei. So now they are attached to the one end. So now as the cavity is enlarging, the outer cells forming the trophoblast will get the flattened. So this is the trophoblast. Now these trophoblasts, they are lined by the flattened cells. And now what is happening is that the inner cell mass, that is called as the embryoblast, they become very compact. Embryoblast, they become compact and they are attaching at one pole, and that is called as the embryonic pole. So embryoblast aligned at one pole, aligns at one pole, that is called as the embryonic pole, that is called as the embryonic pole. So this embryonic pole. Now after this one, what happens is that the hatching as the happens. So the shedding of the zona pellucida, finally the zona pellucida is got rid of. Okay, so now due to which the implantation will be happening. So after this, the fate of the blastocyst will be, first of all, embryoblast. The embryoblast will be giving rise to the embryo, and trophoblast will be giving rise to the placenta, and the blastocele, that is the cavity. Okay, so that cavity is called as the blastocele, and it forms the yolk sac. It forms the yolk sac. So this is how the fate of the blastocyst is defined. Now what is happening in the next video, we will be seeing the implantation. So before that one, let us try to understand what are all the different abnormalities happens at this level. So first of all, the significance of the capacitation. So first, it has to be understood that whether it is required for the fertilization. Yes, capacitation is definitely a prerequisite for the fertilization. So before ending this video, let us try to understand the different clinical correlations of these points what we have learned till now. Okay, so first of all, the capacitation is definitely and a mandatory prerequisite for a oocyte to be fertilized by the sperm. Now what is happening, while explaining the capacitation, I have said it will be happening only in the female genital tract. Now what about the in vitro fertilization is that during in vitro fertilization, the ejaculate is treated with a solution. What does it contain? Mainly the gamma globulin. So the gamma globulin, and then the free serum, and then the serum dialysate. So there is a solution which contains these, mainly gamma globulin, free serum, follicular fluid, serum dialysate, and an adrenal gland extract will be will be mixed to form a solution, and this solution is used to capacitate the semen sample received. So capacitate the semen sample. So that is the first clinical correlation we will be knowing. And then after this one, in vitro fertilization. So basically the in vitro fertilization involves, to sum up simpler words, stimulation of the ovary, so where the gonadotropins administration will be done, and so that the follicular growth is stimulated. After that one, the retrieval of the oocytes. guided by the ultrasound visualization or by the laparoscopy. And then the collection of the sperms, the ejaculate is collected, and then fertilization, or how it is that oocytes are placed in the culture medium, and then the culture of embryo, and lastly embryo transfer. Okay, so the IVF procedure: stimulation of the ovary, retrieval of the oocytes, and then collection of the sperms, and then fertilization of the sperms, and then culture of the embryo, and lastly the embryo transfer. So at this level, let me say you a point: at least 3 8 stage cell embryos will be transferred into the uterus to develop to the full term fetus. Why? Because due to the low success rate of the implantation. So this is how the clinically correlation of the IVF. And now after this one, there is an important question here, that is the hydatidiform mole. So let us end this video by learning about the hydatidiform mole. So what is actually the hydatidiform mole? So hydatidiform mole, this occurs due to the abnormal development of the trophoblastic cells formed. How it is done? So is an there is unfortunate in some unfortunate conditions there will be an empty oocyte which will be fertilized by a sperm. Okay, or due to the fertilization of an empty oocyte by two sperms. So how is that formed? Is that fertilization of an empty oocyte of an empty oocyte. So how? With one or two sperms, due to which there will be generation of the, if it is one, that is the monospermic mole. If it is the two sperms, that is the dispermic mole. Okay, and due to which what happens is that the death of the embryo happens. And as a result, due to which what is happening, the chorionic villi will be not undergoing in a normal condition. Chorionic villi fails to develop and undergo the degenerative changes. So chorionic villi fails and undergoes the degenerative changes, and the degenerative changes are subsequently resulting in the formation of the cystic swellings, which gives a grape bunch like appearance, cystic swellings, and these are called as the hydatidiform moles. And what do they do? Most important feature is that these cystic swellings, they secrete high amount of the HCG, high amount of human chorionic gonadotropin. So clinical features, they are the preeclampsia will be there, elevated levels and enlarged uterus, and then no fetal movements, no heart sounds. Very important symptoms is that the no fetal movements and no fetal heart sounds. And now ultrasound scan reveals the snowstorm like appearance, and that is how the hydatidiform mole is be seen. Okay, so let me show with the diagram. So this is the uterine cavity, and these are the fallopian tubes, and here this is the uterine cavity. And now this entire uterine cavity are filled with the numerous fluid filled cysts. So why? Because due to this fluid filled cyst, it got the name, it gives an appearance, that is the bunch like grape appearance, and this is called as the hydatidiform mole. And it has to be operated by a procedure called as the dilatation and curettage, abbreviated as the D and C. What is happening is that around 3 to 5% of these moles may undergo the malignant change, and they will be resulting in a condition called as the choriocarcinoma. So this is how the hydatidiform mole will be changing. So in the next video we will learn about the implantation, abnormal sites of the implantation, placenta previa, and then the subsequent development of the zygote or the fetus. Thank you.